My name's Jennifer Hamilton. I was at Mount Sinai as a graduate student from 2013 to 2018. I was just enraptured by this microbiology department. It drew me to Mount Sinai. So I spent between 4 and 5 years there, and I got really good at engineering viruses that could kill cancer cells. At the end of my PhD, and I ended up joining the laboratory of Dr. Jennifer Doudna at the University of California, Berkeley. She just won the Nobel Prize in 2020 for chemistry for her development of CRISPR-Cas9 as a genome editing tool. And so I'm in her laboratory now. It's been a little bit over 2 years, and I'm directly applying things that I learned at Mount Sinai and viral engineering to try and develop new, novel, and safe and effective delivery vehicles, both inside the body and to cells outside of the body. It makes it sound like it's a very linear progression from point A to point B, but everything that I learned at Mount Sinai really prepared me for any sort of challenge that I've met in my postdoctoral career.
Mount Sinai kind of helped me build that kind of way of creative thinking, how science touches our lives on a day-to-day basis. And so that got me thinking, well, startups do exactly that. We are innovative, we push the boundaries, we take the risk to try to answer questions that nobody knows the answers to. So that's how I ended up looking for jobs in startups. Currently, I'm working as the principal scientist for a small startup company called Yes Technologies, where we are studying the sense of smell. We're trying to develop a platform technology to digitize the sense of smell, using the sense of smell to diagnose and track diseases. And so one of those candidates for our pipeline is studying Parkinson's disease. My career is still very much science-oriented. It's been 3.5 years and I still love my time at the bench. And that was the same when I was at Sinai. And it's all about how to think about your challenges. If you have a roadblock, how am I going to solve this in an innovative way? How do I think outside the box? And those are the skills that you bring and learn with when you are at Sinai. It sticks with you for the rest of your career.
My name is Michael Letko.
My name is Angela Ho.
I'm Lauren Friedman.
I'm currently an assistant professor at Washington State University, and I was at School of Medicine at Mount Sinai from 2011 to 2015 as part of the microbiology MTA.
I graduated in 1998 with a PhD in neuroscience. I am currently an associate professor at Boston University in the Department of Biology. Our lab is examining synaptic mechanisms underlying neurodevelopment and Alzheimer's disease.
I completed my PhD in neuroscience at Mount Sinai in 2014, and I'm currently at a nonprofit called Alzheimer's Drug Discovery Foundation.
You can try to go for a name of an institution or a certain lab, but I think with Sinai, what did it for me was the whole department, really. So that was what pulled me there, was the microbiology program, and some of the incredible scientists that passed through those halls are what brought me there.
What I've gained as a graduate student at Icahn School of Medicine was to be a strong critical thinker.
I review drug discovery proposals, we make funding decisions, and my training at Mount Sinai helped me to assess the best science, what's needed to answer certain scientific questions, how to poke holes in certain ideas and ensure that there's real evidence behind things that I'm looking at.
My name is Francesca Cole. I was a PhD student in the developmental biology department and graduated in 2003 with a PhD in the regulation of midline development and skeletal myogenesis and the role of the protein kiddo. From my developmental biology PhD at Mount Sinai, I became really interested in how developmental defects occur in humans. I did a postdoctoral fellowship at Memorial Sloan Kettering Cancer Center. From there, I started my own laboratory as an assistant professor at the University of Texas MD Anderson Cancer Center. And I've continued to study how chromosomes missegregate and lead to aneuploidy diseases like Down syndrome. My PhD supervisor was Rob Krause. I have a lot to thank Rob for. I think that I really got some wonderful support, and I think I developed a lot of confidence in myself as a scientist and in what can be done. that you can start with a blank slate of a question and take it all the way to a mechanism.
I had a really great swath of mentors while I was at Mount Sinai, and they were the ones that actually really motivated me to start this company. I completed my PhD in 2019, and my PhD was focused on using machine learning and computational chemistry approaches to characterize protein kinases and create new drugs for them. And that's what I'm doing now. I started a company called AI after I finished my PhD that's focused on creating a new generation of kinase drugs using machine learning.
I graduated from Mount Sinai in May of 2014. I was in the Genetics and Genomics MTA, and currently I am the CEO and co-founder of UVA, a women's health company that actually spun out of Mount Sinai Hospital.
I think Mount Sinai was a really wonderful place to get my feet wet. Actually, before I even started graduate school, I did my undergrad in philosophy, so it was a very different career trajectory, but Mount Sinai really helped me build up the tools to start this company and gain confidence to lead it going forward.
I remember at the end of my PhD, we sat down, we're like, okay, so what do you want to do in your life? And I pitched this idea of UVA to them, and they were like, Amy, do it. See if it works and do it. And Mount Sinai gave me that confidence to actually pursue this really difficult trek of a journey that I've started for myself. I don't think I would be where I am today without their support.
They're really there to facilitate you as a student, but also carry you forward as a scientist and in your future career. Those are the things that are really critical to where I am now, I think.
I'm Yonit Levin. I would say I'm a macrophage biologist due to my experience at Mount Sinai. When I started my PhD, I knew I was very interested in innate immunity and antigen presentation. And so I decided to join the laboratory of Miriam Murad, who is an amazing scientist. I harnessed that excitement and some of the early work from the lab of Miriam Murad to continue to study and really define the role of the tissue-resident macrophages, both in the steady state and then in human tumor lesions. The thing I gained most from my time at Mount Sinai is definitely passion and enthusiasm. The close relationship between the basic science and the ability to bring our knowledge of innate immunity to really understand what was happening in our patients.
I lead the Translational Disease Modeling Group in Rare and Neuro at Sanofi, a pharmaceutical company. We develop these computational models of disease and drug response to inform decision-making in R&D. And so much of what we do requires this very keen understanding of the biological processes of the disease and the pharmacology. I did my PhD in the lab of Rabia Yengar at Sinai, who at the time was one of the few researchers that were developing computational models of intracellular signaling and really pairing them with mechanistic experiments to understand the biology. So that kind of environment really prepared me for what I'm doing right now, really extracting quantitative data from experiments, distilling it to the mechanistic information that can be used to develop these type of models. And I think it really prepared me for a career in science in a variety of different ways.
What ends up being translatable ends up being a lot more than you would think when you first start. I am a research associate and analyst, and what we do is we are an investment bank that then provides research and investment recommendations to our clients, including hedge funds, mutual funds, pension funds. And so we will follow a number of companies that are in our space, whether they be pharmaceutical, biotech, or biopharma companies. It's been quite the environment to be successful as a scientist. I would say that you get a very robust experience in not just your PhD project, but really getting an understanding of what it takes to be successful, how to be a leader, how to troubleshoot or work through problems. And a lot of that resilience that you gain in your PhD is something that is translatable across different fields. I was just kind of reapplying what we've learned in another field.